question, so I'm out of the hook.
You are done. Thank you. Great job. Yeah.
Thank you.
Okay, Chris wants to pull up the slides now, and we can get started with him when he's ready.
I wouldn't like her neither.
Oh, who's next?
You are next.
Am I next? Ah,
You are next. I already did introduce Dr. Evans.
yes. So you've done the introductions. So
And this talk here is entitled Genetic Molecular Approaches to Drug Addiction. Thank you, Chris, for joining us again. We're very honored to have you.
see all my emails. Okie dokie.
Groupon is great.
Okay, so this is the second one. So this one will be much more concerned with genetic tools, and I've tried to give you some idea about the ones we've thought about over the years. And it's not just mice. we've thought about other models, and I sort of wanted to go over some of the other models. So I personally think that using evolution and looking at different species is a very powerful way of doing research, and one should, if one can, sort of use the lowest species possible to answer the questions you need to answer. And with regard sort of addiction and processes regarding addiction, there's lots of very primitive species which can be very powerful for answering some of the questions. So, if you look through evolution, C. elegans are probably some of the first species which have dopamine in them and have a functional dopamine system. And there's a very, very active C. elegans research groups which use these as a model, and they're incredibly powerful. With C. elegans, I think they have 8 dopamine neurons in their system. They just have 8, and I think they have like around 300 neurons total. But the power of these little worms are enormous. You can do screens. You can know every, they know every synapse that these worms make. They've done electron microscopy throughout the whole worm, and you can figure out exactly which synapse could be involved in what function. So they're incredibly powerful in many ways. But what can you do with them, right? You can't do many behaviors with them. Kabir was talking about pretty complex behaviors, even, asking them to remember whether they were in one place or another place. The self-administration you can't really do, but you can do some very simple experiments with C. elegans and ask them, what are those dopamine neurons for? And it turns out that they are involved in seeking food, which is one of the primary things you need to do, is to eat and to find nutrition and as well as replicate, right? So, but to be alive, you need to find the elements that you need to thrive and to make those cells work. So getting food is a real big one, and dopamine right from the get-go is involved in food, searching for food. And if the food is scarce, they'll change their way of moving so that they optimize to come across food. And this is dopamine dependent. And as species evolved, then different things come in. So We have, with our mice, we have many more sophisticated behaviors which we can measure. But, you know, even before that, we have the flies. And, you know, there's been some very elegant work with Drosophila on systems which are involved in addiction, especially alcohol. So and I'll show you a little video a little bit later on the use of flies in alcohol research. But we've mostly gone to the rodents with mice, because we have pretty sophisticated behavioral assays that Kabir has talked about, a few of them, and self-administration is a big one that we use a lot, and pain assays, so we can look at pain because we're interested in the opioid system. So there's lots of assays we can do with rodents. And, you know, coming to the human work, I mean, really all we can do is association studies. And, you know, there's no manipulation of genes. There's no ways of telling, you know, hey, you reproduce with this woman and we'll look at your offspring. You can't really do that with with humans, and it's never going to happen. So, you know, the only thing you can look at is associations, association genetics. And they have lots of problems because most genes, at least with addiction, have been shown to have very, very small effect sizes in the actual addiction process. And as I show you, that most of those genes are really for specific drugs, and they're protective because they cause aversive events. So that's I'll talk about in a minute. But if we look at this sort of evolution, we can see that we've got you know, there's different things we can study at different levels, and the C. elegans have their place, the Drosophila have their place, the mice have their place, and with humans, but we can only study specific things in different species. So I've done quite a lot of work now with the opiate system, and it sort of stops around the vertebrate expansion, which is, you know, maybe 500 million years ago. That's when the opiate system seems to have come into play, and with the vertebrates. And this is where, you know, opioids are involved in stress responses, they're involved in anxiety, they're involved in reward, they involved in gut transport. And these sort of functions are sort of important for vertebrates, but not so evident in the invertebrate system. So it seems to be a system which has sort of evolved from the vertebrate lineage onwards. And that's been sort of some of my research is sort of following back some of these systems, and the opiate system in particular. So we just published that the first sort of opiate receptors that can be found are in these sort of slime fish. They're basically these fish which, well, they aren't fish really. They're notochordates, called hagfish, and they live about 1,000 feet down in the ocean. And we've isolated all of the opioid systems from these fish. And that's the first, they're the earliest species we can find the opiate receptors. So, and they're very similar to the ones in human, very, very small differences. So I've been interested in that. And I'm not sure whether we can use them as a model. These things don't do much, they just wriggle around. So having them doing any behaviors is going to be kind of impossible. But anyway, so if we, as I said, if we look at the different genetic models, we've got the C. elegans and the Drosophila, and the power of these models really is you can do random mutagenesis or targeted mutagenesis even, and you can look for behavioral changes. And that's really, really, really powerful. So you can just do a random mutagenesis over the whole genome, look for a change in behavior, and then figure out what genes changed, and then go back and figure out this gene. And as I show you in the case of Drosophila, this was used to isolate a number of genes regarding alcohol sensitivity. One of these mutants was called cheap date, which I think is kind of insulting to some people now. So I think it's going to get changed, because there's a whole, there's some really quite sort of provocative names that the Drosophila research people have used. So they're sort of, they're all changing now. And but this protein was involved in the sensitivity to alcohol. So that was one of the genes which was found doing these mutagenesis screens, but lots of other very important genes in development and in synaptic plasticity, etc., have been found using these random mutagenesis screens with C. elegans or with Drosophila. So with mice, we can't do random mutagenesis because we'd have to use too many mice to do that. But you know, Nick earlier talked about using different strains and then following genetics around to decipher what behaviors were following what genetic piece. And that's a very powerful way of doing this, because you can actually choose which mice to mate with what. Unlike with humans, you can't do that, right? So with mice, you have a strong you have a strong way of doing both targeted mutagenesis, which I'll show you how it happens in a second, and genetic associations. So mice are really good models. And the reason why mice were so good were that they had these little embryonic stem cell lines, these cell lines, which you could actually do the recombination in mice and start making the knockouts. So that's why really mice have been chosen for many of the genetic studies that they have been, and not rats. But now rats are coming into play because you can now use a new technique, which I'll show you a quick video of, called CRISPR, which enables you to change the genetic material almost of any species. My guess is that people will be using CRISPR on people soon, and we'll be able to change things like genes for in the immune system which are wrong, or maybe improve them, et cetera, et cetera. So CRISPR is going to definitely become useful, and viral technology, of course, is already being used as for the vaccinations. So things are happening in humans where we started expressing new proteins, et cetera, et cetera. So there is some genetic work now for curing some diseases. So that's what I really wanted to say there. We've got all of these different models, and lots of the questions have to be tailored to at different levels, and all of these have been contributive to our knowledge of genes involved in addiction. Okay, so I wanted to start with really the human case. You know, some of the most powerful genes we know of, or most of the ones we know of, are really the ones which cause aversion to specific drugs. So if you have different versions of alcohol dehydrogenase or aldehyde dehydrogenase of which as part of the metabolism of alcohol, if you build up acetaldehyde in the bloodstream, you get the flushing syndrome, and that's pretty aversive, you know, and this is quite protective against alcoholism because it's aversive. It's not nice. And disulfiram, which is one of the drugs which inhibits aldehyde dehydrogenase and builds up acetaldehyde is actually used as a deterrent for alcohol taking. So, you know, this is not really anything to do with addiction. It's due to creating an aversive state of the drug, that particular drug. And the same kind of thing happens with the acetylcholine receptors for nicotine, right? So there's the A5 subunit, which is involved in some of the aversive effects of nicotine, as expressed in the habenula and the interpenducular nucleus, and this is aversive. This is when increased concentrations of nicotine become aversive. And there's some really elegant studies by Christy Fowler and Paul Kenny, which showed that if you take away this receptor, nicotine administration, self-administration, they had a nice little self-administration assay for nicotine, goes up. And this receptor subunit was actually acting as a brake because it's part of the aversive features of, or effects of nicotine. So I always thought that's quite interesting that the most of the pertinent associations with genes that have been found so far are really to increase the aversive state of addictions, not really, you know, this sort of addictive state. And that's what, you know, people have been looking for magic bullets, but I very much think that there won't be one. There'll be lots of little contributions from many different genes which metabolize things, which cause impulsivity or changes in the ability to take risks. Maybe these are endophenotypes which contribute to addiction as a whole. But my guess is it's going to be really complex, lots of little elements adding up. And then on top of that, you have the environmental factors, which are clearly important, things like child abuse and what your partner's doing seems to be very important. If your partner's a smoker or a drinker, so there's sort of bonding, environmental conditions seem to be very important for certain drugs. So it's going to be a very complex. It's going to be very complex to work everything out. But there's certainly going to be some genetic factors, some environmental factors, and these are sort of merging and interacting in such a way to promote the addiction state. So let's have a look at C. elegans first. Yeah, there's around 300 neurons and just over 50 glial cells, actually, males have a few more neurons and a few more glial cells. I don't understand why. There is a big difference here in the number of neurons between males and females there. And there's only about 1,000 cells in the body of these worms. So quite a few, you know, about a third of those are neuronal, which is pretty amazing. And as I said, there's a lot of work on the analysis of this model. So they've done electron microscopy through the whole animal, and they know almost every synapse there is and what connects to what, which is amazing and really gives you an amazing model to study. And they also know what messages are in each one of these cells. So they have a map, a genetic map everything in these animals. And as I said, there's only eight dopamine cells in the head ganglia. I think there's a few more in the dorsal part here, but the in the tail there. But these cells are definitely involved in food seeking, and some beautiful work to show how if you obliterate these dopaminergic cells, you lose the ability to search for food effectively. So dopamine right at the beginning involved in food seeking, and not necessarily reward, but in food seeking. Maybe it's been hijacked for the reward, food reward. Okay, so I hope this works. Yeah, it should do. So this is example of how fruit flies have been used, and it's a little video from a very. Can you hear it? All right.
I know what you're thinking. This reminds you of the
Yes, we can hear it.
Drosophila, right?
Yeah.
Well, actually, it does. If you're neuroscientist and fruit fly expert Ulrike Heberlein at the Howard Hughes Medical Institute's Genelia Farm Research Campus near Washington, D.C. Do you love studying fruit flies? Do you love fruit flies?
I love fruit flies, particularly drunk fruit flies. Quite honestly, I didn't know what we were doing back then, but when I saw the very first drunk fruit fly, I said, this is it.
Dr. Heberlein and her team use fruit flies as a model for studying alcoholism. When the flies breathe ethanol vapor, they get hyper, bump into the walls, become unable to fly, and then pass out. So what you really saw was the arc of a human buzz.
Absolutely.
In something that buzzes.
It's very, very, very good.
Here they're homing in on specific addiction genes. Mutant fly strains are named Cheap Date, Happy Hour, and Tank, depending on their genes and how they control the hankering for booze. The researchers are also looking at how outside influences can encourage flies to become bar flies. After male fruit flies are spurned by females not interested in mating, they drink twice as much alcohol as those who just had sex with a receptive female.
They may compensate by drinking to normalize their sort of reward system.
Key to the reward system for fruit flies and humans are molecules called neuropeptides. They travel from neuron to neuron, allowing brain cells to communicate with each other.
Rejection reduced the levels of neuropeptide F, and mating increased the levels. And we were able to show that reducing the levels of neuropeptide F leads to enhanced drinking. The converse, elevating levels of NPF, led to reduced drinking.
But neuropeptides are just part of the internal chemistry of reward, and it's linked to addiction.
Okay, there's Nora Volko doing her walk and her exercise to keep her reward system healthy. Okay, so if you let's go on to the next one. Okay, okay, so yes, so definitely flies have their place in research, and there's some very interesting genes which can be plucked out of those flies by using these screens. That she was using those canisters, and she would put vapor, alcohol vapor in those canisters, and she would basically see which ones flop down first. And then she'd after making mutants and just random mutants, and then she'd look for the ones which weren't able to, which were intoxicated very fast with the alcohol, and then she'd analyze those mutants, and that's how she was able to isolate Cheap Date, which was one of those animals which just flopped down very, very fast with with a low concentration of alcohol vapor. So, and now she can start doing other experiments with seeing, you know, with what happens when the fly who wasn't satisfied mating was taking more alcohol, and then looking at the circuitry of this. So there's lots of things you can do in flies, and they shouldn't be disregarded in addiction research. They certainly have their place to offer in identifying some of the molecules involved. And whether that's going to, you know, pan out to human research is a whole nother story, but that goes for any of this preclinical research. There's always issues of translation. So my interest has always been the opioid system, and I spent a lot of time trying to get the nuts and bolts of this system during my career. I started isolating these endogenous opioid peptides, and we found a few new ones. Dynorphin 128 was one of them, and one of the metorphamide was from this proenkephalin precursor. So they're basically made from these opioid precursors. They're chopped up by very specific proteases in the dense core vesicles. These prohormone convertases and amidating enzymes and enzymes which chop off the C-terminal basic residues. So I spent a lot of time in this area, and as I said, we identified a few of these new opioid peptides. There's about 30 different opioid peptides which are made in the brain, and they come from these three opioid precursors. Kabir actually was talking about the system which orphanin FQ system. This is the ORL1 receptor he was talking about. This is very much like an opiate receptor. It's certainly part of the same family, gene duplicated. They all have the same sort of structure, basic structure. They all have an intron-exon boundary here. They have sequences in common. Very all homologous precursors, and the receptors are also homologous, very high homology between the different opiate receptors. But of course, as I told you earlier, they do very different things in the brains because they're on different neurons. So we know that the delta opiate receptors involved in depression, anxiety, the agonists are anxiolytic. Delta receptors are also involved, seems to be involved in chronic pain states. They're not very good with acute pain, but they're very good at chronic pain. The mu opiate receptor is the big one, which is the target, the primary target of fentanyl and all the opiate drugs that we know about and are used therapeutically for pain. So, and the Kappa receptor is kind of an interesting one because most of its effects are exactly the opposite of the mu opiate receptor. It's the target for salvia, which is that hallucinogen in magic mint, which is, I talked about a little bit earlier. We know a lot about this receptor, and we've done a lot of work on this in the lab. This is upregulated with chronic pain states, so it may contribute to some of the, it looks like it does contribute to some of the aversive states that happen with chronic pain or after withdrawal from drugs. So this is a sort of a very sort of activated receptor system. It's found actually, it's the major precursor for this is dynorphin, and dynorphin is actually shown to be upregulated in suicide victims. So it seems to be one of these targets which may be important in the adaptive states to chronic drug use or chronic pain, the aversive, the dysphoric states which occur with chronic drug use. So we're really interested in this, and there's a lot of interest in developing kappa antagonists which are for treating this as antidepressants, as anxiolytics, as and we're sort of interested to sort of blocking the dysphoric phases of drug withdrawal. Now there's potential problems with drugs like this because they can also be used to enhance the effects of other drugs of abuse. So Kappa antagonists will will enhance the effects of cocaine because Kappa receptors, which, as I said before, turn down neurons, they're on the dopaminergic neurons. So cocaine is relying on the release of dopamine. Kappa is going to actually stop the release of dopamine. And so cocaine's not going to work so well because it hasn't got so much dopamine to block the reuptake of. So, these receptors, very important where they are. And since we cloned the delta receptor, these other receptors were so homologous, they come out very quickly. So now we have all the probes to study these receptors using in situ hybridization, which Nick talked a little bit about. We use fluorescent techniques now, RNA scope, which is a way of looking at multiple messages in the same neurons, and is very, very powerful. So we don't have to use radioactivity anymore, which is nice. I hate radioactivity. It's always a bother. But these fluorescent techniques now are very, very powerful, and you can actually look at multiple messages in the same cells, up to about 6 or 7 messages in the same cells. So now we can look at the co-localization, say, of GABA, a GABA cell with an opiate cell, or a Kappa with a mu, et cetera, which makes very powerful techniques. Okay, so this is the opiate system. 4 precursors, 4 receptors, not always mixing and matching. The proenkephalin looks as though it's makes peptides for both the mu and the delta, prodynorphin, the kappa, and the orphanin FQ, activating the SOR1 receptor. These are G protein coupled receptors, as I said before, which go through the membrane 7 times, and the homology between these receptors, these OPA receptor families, very, very high. I think the red is between 80 and over 90% homology in this diagram. Okay, so what do we use the mice for to study? And because this is our primary model, and we've really used this as to understand the opiate system, which receptors are involved in what, and I'm going to show you some of the data through the years, mostly from my collaborators, Brigitte Kiefer, who cloned the receptor about the same time as us and went on to do a lot of mouse genetics afterwards. So one of the first ways of manipulating mice was to generate these knockouts. As I said, these ES cell lines were really, really important, and really sort of drew mice into the first to being the genetic models, because of these ES cell lines were easy to inject, and you could get recombination of genetic material by injecting targeting vectors, and the genetic material would recombine, and you could get knockouts generated using targeted mutagenesis. You could also inject additional pieces of DNA with big pieces like bacterial artificial chromosomes, which were expressing specific genes. So we've used those techniques as well in the labs, in our center, and that's worked very nicely. And I'll show you one experiment where we use that. But the idea is that you inject these blastocytes into the mouse, and you generate offspring, the chimeric offspring. Then you have to breed them up, and about a year later you have mice which you can use for research. CRISPR is a much quicker technique now, and people are using this, and it can be used for many different species. So CRISPR is going to be the technique of the future for generating mouse models and rat models and guinea pig models, or whatever model you want of whatever species you want. So that's going to be the technique of the future. And I have a little video showing you about CRISPR in a second. Okay, so if we look at the knockout mice, these were just plain knockout mice. You can see that, and this is autoradiography using a mu ligand. So you basically just cut a section and incubate it with a radio-labeled mu agonist, one which only sees the mu opioid receptor. I think this was Damgo, which is a peptide, and you can see it binds to certain places in the brain. This is the wild-type mouse, and this is looking at the mu ligand, and you can see that you get a beautiful staining. This is the heterozygote, which has one allele of the mu receptor and one knocked out. See, there's about half the number of receptors. And this is the knockout. So you've just taken away the mu opioid receptor from every cell in the body. No mu opioid receptors left. So if you look at what happens with this mouse, the reward for morphine has gone, so there's no place preference, which Kabir showed you with opioid drugs. They don't care whether they get morphine on one side or the other side of the apparatus. They don't care. They just don't care about morphine anymore at all. So that's gone if you take away the mu receptor. And obviously dependence is gone. There's no opiate dependence, and just no activity at all with morphine. With delta agonists, there was a few changes, but the important things here was that also other reward activities were gone. So the reward for alcohol was gone. The reward activity for cannabinoids had gone. So it seemed like this endogenous system, some circuitry with the mu opioid receptor was involved in the rewarding effects of both alcohol, THC, and it's also shown for nicotine as well. The opioid receptor was an important component of the pathway for rewarding effects of these drugs. So with these mice were created by Brigitte, our collaborator, and we've used them in a number of tests. We've also have knockouts for the delta, the Kappa, and the ORL1 receptors. The interesting one is really the Kappa receptor knockouts. And if you know that, high doses of cannabinoids are usually aversive. And if you look at the Kappa knockouts, they don't show that aversion with high concentrations of THC. So they seem to have lost some of the aversive components of THC. So they're pretty interesting. The effects of morphine dependence, the withdrawal effects seem to be also decreased, and the analgesia and dysphoria of Kappa agonists have gone. But the morphine dependence also shows decreased effects in the Kappa antagonists. So here we have some,
Chris, does that mean that THC binds to THC
No,
binds
no,
the
no,
Kappa
no, no, no,
receptor.
no. THC is high concentrations of THC show a place aversion, like Kabir was showing in his mouse assays. So high THC shows an aversion, but if you take away the Kappa receptor, that aversion isn't there. Doesn't mean that THC is binding to the Kappa receptor. It means that the circuitry involved in THC aversion seems to involve the Kappa opiate system. So I don't know if anyone's done that with the dynorphin knockouts. Actually, that would be the way to show that there's no direct interaction, but it requires the release of dynorphin to activate the Kappa opiate receptor. I can't remember if anyone's done that or not, but the interpretation is that the Kappa system is activated with by THC, not that THC actually binds to the Kappa opiate receptor. Okay.
But as you mentioned, Chris, I think it's most of these drugs of abuse over time, they increase the level of dynorphin. Amphetamine
they
does,
do. They
all
do.
those. So most likely it does. And we have dynorphin knockout mice, but we have, other projects. Probably we can try.
Yeah, you should try that, Kabir. Just to see if the dynorphin knockouts show the same as the. It's an easy experiment. It's just a place. It's just a place of aversion with a high concentration of THC, which I think is easier to get now because of the laws of changing, and THC is more available, which is good. So anyway, it's
hello, Dr. Evan. I have a question here. In this slide, you're showing mu receptor mutant mice. The delta expression is increased in these mice?
Maybe a tad. Maybe a tad. There was a big argument in the field about whether it was significant or not. This one sort of implicates that it might be increased, but it isn't really clear. It isn't really clear that there wasn't a major increase in the delta. Now, if you take proenkephalin out, there's an increase in the mu, definitely, because there's a constitutive downregulation of mu opiate receptors. But I don't think there was a big compensation. There isn't a big compensation with the delta if you take away the mu. Okay.
Chris, does it have to do anything with the dimer, the mu delta dimer, maybe?
Yeah. Yeah, but there wasn't big changes, Kabir. There wasn't really big changes. I mean, there wasn't really big changes in that. It was marginal when they did the statistics. It looks like in the heterozygotes, it looks a bit more delta, right?
Yes.
In this ones, but not in the homozygote knockouts. That looks more, maybe a little bit more. But I remember there was a big discussion about that in the field, and it was, if there was a change, it was considered really quite minimal. I had to change my settings here to see that. Okay, let's go on. Right. Okay, so well, some of our questions have been, and I thought this would be a good way of explaining how we've been doing things, is which opiate receptors on what cells trigger their reward and antinociception, and which may be involved in withdrawal. So what we first did when we started, and we first got the sequence of the opiate receptor, we were able to make antibodies for once. So we made antibodies to the mu opiate receptor. And this is the distribution. The white is showing high concentrations of mu-opiate receptor. And as you can see, they're in these patches of the striatum, which is really a hallmark of the mu-opiate receptor. This is a rat brain. And you can see there's high concentrations here. So the implication is they're involved in reward in the striatum. They're in the hippocampus, they're in layers, I think 2 and 5 of the cortex. They're also in the dorsal horn, where they may be involved in pain mechanisms. And so you can see there's a distribution here. They're in lots of different places in the brain. So the questions we all had was, which ones are important for reward? Which ones are important for pain? And that's where you can start using knockouts and conditional knockouts. The nice thing about the opiate receptor is, you've got really good drugs to study it. So some of these studies were performed by doing micro injections into different parts of the brain, and this is what has become pretty complicated. So if you look at reward, for instance, you can get rewarding effects by injecting opiates in many different parts of the brain. You can also get analgesia in injecting opiates at the spinal cord level, in the PAG, in areas of the cortex. You can get analgesia. So there's, we don't think of the opiate receptors as having just one locus of action. They can mediate analgesia in many different parts of the brain. So anyway, but our question was, what opiate receptors may be involved in reward and antinociception. And I'm going to show you some of the studies which sort of speak to that. So one other technique which I should tell you about, antibodies are a pain they're real pain. They often show false positives. We've had antibodies. Our monoclonal antibody to the ORA1 receptor looked like it was a beautiful antibody, stained exactly how we would expected it based on the messenger RNA levels, the in situ hybridization levels. But when we looked at the knockout, which took out that piece of receptor that we've made the antibody to, turned out that it wasn't actually binding to this receptor. So it was a bad monoclonal. But anyway, this is a way that you can actually use genetics to show up antibodies. And what Brigitte did here is she took the delta and the mu receptor, and she tagged the delta with green fluorescent protein and the mu receptor with M cherry. So these are two different fluorophores which she made genetic mutants for, and she basically tagged this GFP or the M cherry at the end of the receptor, and then you had these beautiful reagents for studying the receptor in real time. And she has some very nice videos showing how this worked. And you can see the co-localization there. And there really isn't a heck of a lot of co-localization, Kabir, in the brain. There is some in the brainstem, but the delta and mu really didn't look like they were co-localized in a heck of a lot of cells. Okay, so as sort of the genetic tools have evolved, one of the most important systems is this Cre recombinase system, this Cre-Lox system. And Brigitte made this floxed mu receptor. So basically what she did is she put these Cre-Lox P sites either side of 2 of the exons in the mu-opiate receptor. So when this saw this Cre recombinase, these combined and they basically took out the mu-opiate receptor. So it was basically a knockout. There's other strategies you can use. You can basically put a stop codon in the beginning of the sequence, so you can flux the stop codon. So normally the gene is not being expressed, but when you excise it, then the gene does get expressed. So you can basically have a knock-in system where you have Cree expressed. So this system is fantastic, because Cree is this recombinase which is able to specifically recombine the DNA and give you expression or lack of expression in specific cell types. And this has provided us really a good tool. So I think I'll show you how this works. This is a nice little so the Cree recombinase binds, and then it loops in, and then there's a chops off, and so you have this excised piece of DNA, which then recombines your piece of DNA and changes the function of that piece of DNA. So it's just showing you what Cree is. It's a recombinase, and it's really found in bacteriophages, and it was used for recombining in bacteriophages. So really a very nice system which was adopted. And this is the LOXP site. This is the core site, and then it's palindrome each side. So it makes this kind of hairpin loop, which then gets clipped. It's a really cool system. We use it a lot, and it enables us to modify genes in specific cell types. But we have to express the Cree first. So there's lots of mouse Cree lines now expressing Cree in specific cell types, which is very important. Okay, so that basically shows you that. So Brigitte made this floxed mu receptor, and this really has given us the tool to study where, you know, the cell types which are involved in different actions of morphine. So you can either use a Cree animal expressing a Cree under a specific promoter, or so, you know, we have Cree animals which are expressing the D1 promoter, the D2 promoter, the A2A promoter, the you know, lots of different promoters. Or you can use viruses. So you can inject a virus which has Cree, expresses Cree, and then you can get mu expressed in certain brain regions with this with these animals, which is really powerful, because now you can choose a technique to have tissue specific or cell specific expression of mu opiate receptors. So this was one of Brigitte's earliest studies. She used this to take away the mu opiate receptors from these primary afferent NAV 1.8 neurons, and then she was looking at analgesia. So these are primary afferents in the dorsal root ganglia, and she can show that really this didn't have a big effect on the analgesic effects of morphine. This was in these initial assays, these acute pain assays, but actually when she had inflammatory assays, she did start getting some changes with morphine with these mu knockouts. So it was working more on chronic pain assays than it was on acute. So that was kind of an interesting finding. So this sort of gives one example. We've used it for understanding which cell types in the striatum are involved in mu mediated processes. So this kind of started with a collaboration with William Yang in our center. What he did is he made a back gene where he had the dynorphin promoter driving the mu opiate receptor. So remember, dynorphin's found in those is that neurotransmitter is the opioid which activates the kappa receptor, but it's found in the D1. It's co-localized with D1 neurons in the striatum. So D1 receptor medium spiny neurons express also prodynorphin. So he used the prodynorphin promoter driving the mu opiate receptor and he made this, and he fused this with the knockout mice. So there was no other opiate receptors, but receptors in the mu opiate receptor in the D1 containing neurons, which is the dynorphin containing neurons. Yeah, beautiful results there. Was completely able to reinstate the reward from in the knockout animals. So that was pretty interesting. So we, well, you know, Brigitte carried on and did a little bit more work on this. And what she did is she used her Flox mu receptors and she crossed these with a DLX, a Cree, which expressed by this DLX5/6 promoter, which only expresses Cree in the forebrain neurons. So all of these neurons were deleted. All of these forebrain neurons were gone, but she still did retain the mu receptors in the VTA and the substantia nigra and the brainstem. But all of the opiate receptors here were gone. And she still had reward. She still had self-administration. She still had place preference. In fact, it was a little more sensitive than had these been removed. So that was kind of interesting, right? So we put the opiate receptor back into the striatum, and it was able to produce reward. But if we take away all of those neurons, there was still reward. So it was that one of them was sufficient, so we could put mu opiate receptors back in the striatum, in those D1 neurons, and it was sufficient to create reward, but it wasn't necessary for reward. There are other systems in the brain which could manage reward.
Chris, maybe 2 or 3 more minutes.
Yeah, okay, I think I'm nearly finished here. So so what we did then was we took these Flox mice and we've taken them out of D1s specifically, and we've taken them out of D2s. We've taken them out of the interneurons, the cholinergic interneurons with chat-Cree, and we have some very, very nice results there where we can show if we take away the mu receptors from the D1 neurons, we more or less lose morphine-induced locomotor sensitization. We don't have the locomotor effects hardly at all, actually, in these D1 Cree mice. And if we take away from the D2, we have increased morphine locomotion-induced locomotion. So we know at least that the striatum is contributing somewhat. These neurons are contributing somewhat to the effects of morphine. Now, the nice thing is that when you have these Cree mice, well, because Brigitte's now just made a mouse which expresses Cree in mu receptor cells. So now you can do optogenetics, you can do DREADS. Nick talked about optogenetics, but DREADS is a sort of another way of modulating cells, but it's a sort of longer lasting. It's like putting a drug receptor into the cells which express Cree. So that's a really nice technique. And these are designer receptors which will turn off or turn on the cells. So that's another technique like optogenetics, but it's like putting a drug in and specifically activating certain cells. So now we can do this. And then we have CRISPR, and I think this might be too long. I think it's like 4 minutes, so I won't show this, but if you're interested in how CRISPR works, this is kind of a nice little presentation. Basically, it basically shows you that how the system works and how you can modify DNA almost in every species now to make knockouts or knock-ins or mutations. It's a very, very powerful technique, and I think in the future that's going to be the new way of doing these genetic modifications in animals and maybe people as well. Okay, so I'll stop there and stop sharing.
Thank you so much, Dr. Evans. It was a great talk and really exciting techniques. Want to have any questions?
I was just wondering, can you send the CRISPR to Christina so that we can take a look?
Yeah, sure. Yeah, sure I can.
Maybe you can put it in the chat, Dr. Evans, if you'd like to put the link, maybe, if that's possible.
Yeah, I'll try and find the link. I have the video.
Okay. Oh, gotcha.
Yeah, I have the video, but it's kind of a nice little video. It's yeah, I'll find it again. No problem. I know I can find it. I'll chuck it into the link.